Okay, so arthropods have a very clever trick up their sleeve. So their basic body construction is to have lots of segments that are pretty much the same. So if you think of something like a brine shrimp, a crustacean, where all of the limbs are the same, or indeed a millipede or a centipede, where each segment has a pair of limbs or two pairs of limbs, and those continue all the way down the trunk. Many arthropod groups have specialized groups of segments to do particular jobs. And in something like a lobster, not only are those groups of segments specialized, but each appendage, each pair of limbs, looks very different very often from the ones next to it. So there are pairs of limbs in the lobster that are specialized as antennae. There's two pairs of antennae. The mandibles, specialized limbs behind that for bringing food up to the mandibles. Go a bit further down and you've got specialized crushing claws, and you go into limbs, walking limbs that have smaller little claws on them, and then you go down the trunk into the abdomen, and you have swimming legs, so legs that are like paddles go further back down the animal, and you go to a splayed tail fan, and all the muscles in the abdomen can contract, and it's called the charadoid escape reaction. They flip their tail and they zoom off backwards. It's a very clever bit of biomechanics. But the key thing is that they've specialized these limbs for very different functions, and that's a bit like a Swiss Army knife. So there's a given tool for every particular job. So this enables them to diversify into lots and lots of different niches and environments. And of course, the head of an arthropod is also a structure, a tagma, which has evolved by recruiting walking limbs from the trunk into the cephalon, into the head capsule. And you can see the remnants of these limbs. This is the drawing of a head of a cockroach, and you can see the maxillary palp and the labial palp are the remnants of legs. You can see it even more convincingly in this head of a locust here, these palps. And this also gives the lie to the segmentation of the head. You can see that very clearly. Now, in common with crustaceans, hexapods, insects, have a head that's made up of 5 segments. And in crustaceans, you can see those segments very clearly. There are 2 pairs of antennae, the mandibles and first and second maxillae. 5 pairs of limbs within the head. In insects, the second antennae are lost, so it's only the first antennae that you see, and then there's a segment that doesn't have limbs, but it's the same construction of the head, because remember, insects are just specialized crustaceans. That makes the crustaceans paraphyletic. Now, one of the keys to the success of arthropods generally is their incredibly versatile exoskeleton. And the exoskeleton of arthropods is a composite material. You gain the benefits of 2 different materials in making up the composite. So you can have something which is very good for strength and something which provides flexibility, let's say, and you combine those and you get a new set of properties. So there are many composites within biology. So our bones, for example, are composites. We have calcium carbonate within a protein matrix, a collagen matrix in our bones, with calcium phosphate hydroxyapatite, for example, within our bone material. And it's quite often that you have a fibrous material within a more flexible matrix. So things like artificial composites are, of course, very important. And there's a whole field of biomimetics where people look at natural structures and try and learn engineering principles from those. So carbon fibre, these are fibres of carbon within a plastic matrix. And this gives immense strength, but also very, very light material. And one clever wheeze is to orient the fibres in different directions. And that gives you strength in a variety of planes. So you probably know how plywoods work. Wood, of course, has as a natural material, it has a grain in terms of the way that the wood grows. And in general, if you put forces across the grain and try and break the wood across the grain, it's very resistant. But if you take up a sheet of wood and you break it with the grain, then it will tend to break much more easily and fracture. And in a plywood, you take relatively thin sheets of wood in which the grain is very strong, and you orient those at 90 degrees to one another. So you not only have strength in this direction, you also have strength in that direction. And of course, both orientations give you strength at 45 degrees to that. And plywoods are made up of stacks of thin bits of wood glued together, again, really to make a composite. Glue also has its own biomechanical properties, and then heated and sort of made into a laminate sheet. This piece of marine ply. Now, arthropods have the fibres, in this case, are a polysaccharide called chitin, and that chitin is within a matrix of protein. So the chitin fibres, they don't just go at right angles, but if you take a section through a piece of arthropod cuticle, there's a slight angle of each layer. And these layers gradually spiral. So if you take a section through a piece of arthropod cuticle under the microscope, you can see these swirling patterns, which are as the fibres gradually change orientation as we go down through the structure. And this gives incredible strength for remarkable lightness. And that's how arthropod cuticle is built up. And you can also, depending on the ratio of chitin to protein, you can vary the flexibility. And in things like trilobites and in decapod crustaceans like lobsters, some extent in shrimps, so in crabs, not only do they have this chitin and protein, they also have calcium carbonate, which gives, again, a much more rigid and solid structure. Now, if you're going to dice, and if you're going to dice a hard exoskeleton, you have to do that in a particular order. So there are particular suture lines, and bits of the armour are shed in a particular sequence. And that's very important. So the behaviour of the animals is tailored to their particular mode of ecdysis. And of course, one of the downsides of having to shed a massive exoskeleton is that you're losing a lot of material. When we have our skeleton on the inside, and we can remodel our skeleton in response to different stresses and strains that we experience. It's why astronauts lose bone mass in space, because they're not subjected to the normal forces. But we therefore recycle the material within our skeletons very readily. But arthropods also try to recover as much as they can of the exoskeleton when they shed it. And so the first thing is there's a molting gel that begins to dissolve the procuticle, the underlying layers of cuticle, and recovers as much of that as possible. And then those, all the goodness in that, all the goodies, as much of that as possible goes into generating the new epicuticle and procuticle underneath. And so this remaining bits of epicuticle and procuticle eventually fracture and get shed, and the new cuticle is formed underneath. Now, that new cuticle is initially rucked up a bit like a sort of folded parachute, and the animal expands its body by pumping hemolymph into the body cavity and effectively pumping itself up, so that when the new cuticle is tanned, cross-linked and made to go hard, that the animal has increased in size in that way. So the external skeleton of arthropods gives them an external armour. And that in many ways is the key to their success and the key to the ability to specialise appendages, lots of appendages for very specialised tasks. So having lots of limbs, having tagmosis, and being able to modify those limbs are key to their success. They also have amazingly complex sense organs, eyes, olfaction, antennae that are able to smell. Many beetles have richly branched antennae that are important for picking up pheromones and other smells of great importance. And most arthropods are also very small, not all of them, but this means you can fit a lot of them in a given space, and they're also able to exploit physically small spaces that wouldn't be available to many other animal phyla. So you could see this coming, but here we go. So here's a quick bit of classification for arthropods. Principally, there are 5 groups. If we go way back in the fossil record, things become much more complicated, but we're going to stick to the simple taxonomy of 4 living groups and 1 extinct one. So first of all, the extinct one, the trilobites. These are incredibly numerous as fossils, particularly from the Cambrian up until the Devonian, although they eventually go extinct at the Permo-Triassic mass extinction. And we'll see some trilobites in a second. Then there are the chelicerates, so think spiders and scorpions, the myriapods, myriapod, lots of legs, centipedes, millipedes, crustaceans, crabs, shrimps, barnacles, and the hexapods, which include principally the insects. So let's take some of those groups in turn. But before we do, there's one other group. I sort of misled you slightly. These are weird. These are amazing animals called the pycnogonids, or the sea spiders, in inverted commas. Sometimes also called the pantopods. Pantopods just means all legs. And they are all legs. The body, here's a pantopod. The body is somewhere in there. Most of the digestive glands, most of the internal organs are within the legs. So they are all legs. This thing here sticking out the front is the proboscis. So the body is just basically the bit where all the other bits join. Here's one with a slightly more meaty body, but these are amazing animals thought to be variously either somewhere sister group to the chelicerates, and therefore the spider, they're not spiders, but the spider name some of the similarity may be because of morphological homology, and people have pointed to the chelifors, the first appendages of the pantopods, as being very similar to the chelicerae of chelicerates. People like Jason Dunlop have made this point. And other people place them way down at the base of arthropod evolution. Somewhere down just above the onychophorans and tardigrades, sort of sister group to the other euarthropods. So the having them close to chelicerates idea is more based on morphology, and the having them at the base of the euarthropods idea is more based on molecules. But the jury's out, as I understand it, on precisely how these things fit in. But they are an amazing group. So the four main groups then, the four main living groups and the extinct trilobites. Here's a trilobite. It's got the name trilobite because of these three lobes to the body, the central rachis and these pleural lobes. Head here, a thorax and some fused segments at the back end called the pygidium. So normally in a trilobite fossil we only have the mineralized cuticle. So this is a hard cuticle that preserves very well, and that's partly why the fossil record of trilobites is so impressive. Compound eyes, we don't normally see the limbs, but in exceptional cases we see the limbs preserved, and they're biramous limbs. They're similar to the sort of archetypal arthropod design. A lot of these things were scuttling on the bottom. Some of them were predators, some were filter feeders, but there were also trilobites that were able to swim through the water column, had very, very large compound eyes and fairly wide degree of vision, more streamlined shape. Some of them had their eyes on impressive stalks. Some of them had lots of weird and wonderful spines. There may have been sexual dimorphism here. These may have been defensive structures. But occasionally, as I say, you have limbs preserved. This is a pyritized fossil of Triarthros, and you can see the legs underneath the animal. Huge range in size. The biggest were up to about 70 centimetres or more in length, and these were tended to be more concentrated in colder water environments in higher latitudes. And in many ways, the trilobites were doing the job back in the Cambrian and the Ordovician that that was later to be done by crustaceans. So crustaceans, in many ways, when those really diversified, and the decapods in particular, when the decapods diversify in the Devonian, they kind of pushed the trilobites out to the margins a little bit. And the trilobites suffered very much at the end Devonian mass extinction, and then finally bit the dust at the Permo-Triassic. And unless anyone knows differently, it always occurs to me that there might somewhere or other be a deep sea trilobite still alive. That's just a fantasy, though. So this is a rather nice shot of one of these lovely spined trilobites. And you can see the compound eyes here. This particular compound eye has a sort of panoramic view. It's kind of elaborated in the horizontal plane so the animal could have seen more around itself. And there's another shot of a Triarthros specimen. You can see the jointed antennae. You can see the limbs beneath the head, the cephalon, and you can see the biramous two branched limbs. You can see them quite clearly here underneath the body. Again, this is a pyritized specimen preserved in fool's gold. And there we are. That's the fossil record of trilobites. As I say, hugely diverse marine organisms, and huge shower of diversity in the Cambrian going into the Ordovician. Silurian and Devonian declining a bit, nobbled at the end of the Devonian, and then limping on to the end of the Permian. So that's the most fossil arthropods that don't fit into one of the big four modern groups are trilobites. So here are the big four modern groups: the myriapods, lots of myria, lots of pods, lots of legs, things like centipedes and millipedes; the chelicerates, spiders and scorpions; the crustaceans; and the insects or hexapods. And together this group make up the pancrustacea. So let's deal with the chelicerates first, because if you think back to that phylogeny, we said that the chelicerates were the first group to branch off. In common with many groups of arthropods, they've made it from the sea, perhaps in through fresh water, to the land, so they are terrestrialized. And when we think of chelicerates, we normally think of the terrestrial, the land-living ones. But there are marine merostomes, things like the horseshoe crab, and back in time there were giant marine eurypterids, so-called sea scorpions. About 60-odd thousand species of chelicerate described, so they're a very diverse group. The body is divided into two tagmata, and we don't call it the head and a thorax and abdomen. It's the prosoma and the epistosoma. The prosoma is the bit that has the eyes and the chelicerae and the legs on it, and the epistosoma, the back part, typically doesn't have appendages on it, and in spiders at least it's not obviously segmented either. So the prosoma with the legs and the epistosoma with the body. If you ask children to draw a picture of a spider, they'll draw a head with a smiley face, and then they'll draw a body with the legs sticking out. In fact, of course, it's the other way around. The limbs are on the front part, on the prosoma. The prosoma has chelicerae, in spiders they're poison fangs, a pair of appendages behind that called pedipalps. In a scorpion, those are the claws, the pedipalps. In a spider, they're little club, I'll do that again for you if you like, little club-like sort of things. And then there are 4 pairs of walking legs behind that. So spiders, scorpions, mites, ticks and so on. Here is a eurypterid. This one looks like it's got a nice smiley face, but this is of course a sea scorpion, thought by some to be close relatives of the living horseshoe crabs, things like limulus, thought by others to be higher up in the chelicerate tree, but again that's the jury's out on that. And many of them grow to hugely impressive sizes, so Jackalopterus here way in excess of 2 meters long, and in many ways seem to be filling the ecological niches of a very large decapods and also some predatory fishes as well. And here's the living horseshoe crab. You can see the prosoma here with the walking legs. You can see the chelicerae, the little chelate appendages, homologous to the poison fangs of spiders, the pedipalps behind that, and then walking legs in behind here. So 1, 2, 3, 4, 4 pairs of walking legs. And at the bases of those walking legs, you can see the gnathobases, so the chewing surfaces, and the ventral food groove. So all of the walking legs serve to macerate food at the point where they join in the middle here and move that forward to the mouth. So the prosoma here covered with this horseshoe-shaped shield, and then the back end of the animal with flap-like limbs that the animal uses for swimming. So it swims upside down, reclined, sort of beating its epistosomal legs in order to sort of swim itself along a bit like a boat. And this long telson spine at the back end of the animal. And also this is the dorsal part of the animal. You can see these eyes here, made up of lots of facets, so convergently evolved compound eyes in the horseshoe crabs. These are living fossils. So on the east coast of America, particularly around the Delaware Bay area, there are particular nights of the year around new and full moons, when the tides are right, where these things all come ashore and spawn. So the female digs into the sand and lays these rather lovely turquoise colour eggs in the sand, and those then hatch out. But this is a mass mating and mass molting bit of behaviour. And this has been going on for this whole process has been going on more or less unchanged for the last 400 million years or so. This is a fossil Xiphosuran, so a fossil horseshoe crab. You can see how remarkably similar they are to the living ones. I once went to an amazing talk on why horseshoe crabs weren't really living fossils, and it focused on little tiny differences in the shapes of these spines and the number of these spines. Just convinced me that they were probably the best example of a living fossil that we had. Amazing how these animals have been unchanged, more or less unchanged, for hundreds of millions of years. So those are the marine chelicerates, because most chelicerates we think of as being on the land, and this is a spider, of course. Spiders have this amazing ability to produce silk. You can see the chelicerae here, the poison fangs, the pedipalps behind that, and the 1, 2, 3, 4 pairs of walking legs, all coming out of the prosoma, with the opisthosoma behind. The opisthosoma does have appendages right at the back end, the spinnerets that are used for producing the silk and teasing that out. But you can also see eyes, and not just 2 eyes, but multiple eyes. In many chelicerates, in many spiders in particular, there's amazing sexual dimorphism. So this is the female and this is the male. And things don't always go well for the male. So approaching the female is often and mating is often the last thing it will do. Fairly often the female waits until she's been mated, if she feels benevolent towards the male, and then eats him. But this happens very differently in different species. These are one of my favourite animals. These are jumping spiders, the Salticidae, and often they have amazingly bright iridescent colours as well. Beautiful animals with amazing vision, amazing depth perception, and amazing ability to jump on their prey. Mercifully, they tend to be quite small, but you can see these amazing eyes, and these eyes are a bit like telescopes. They extend right deep into the prosoma. And you can also, if you watch a jumping spider gauging distances or looking at you, it will sort of crane its head around and sort of look at you from different angles to get the depth right. But there we are. There's another rather lovely one, and I think probably inspired the Sentinels from the Matrix films as well. You can see these multiple eyes. So that the large eyes are, the 2 at the front, are giving you the most resolution and the best depth perception. The other eyes are giving an all-round field of view. But there are other groups of arachnids now, so not just the spiders. The other groups of arachnids, you'll notice, again, the limbs join onto the prosoma. The opisthosoma of spiders, of course, doesn't show any segmentation. The opisthosoma of the other arachnid groups does, much more visibly. So none of these are spiders. This is a thing called a Europygid. This is an amblypygid. Notice how the limbs are modified. The pedipalps and chelicerae are modified in different ways in different arachnid groups. This is a sun spider or wind spider, solipugid. These enormous, great big chelicerae here. This thing's horrible. It has a rather sort of squishy looking body and anaesthetic saliva. So it's able to start grazing away on things it wants to eat. And there are these rather nasty apocryphal stories of people waking up with them just grazing away on the side of their face. Horrible. And a scorpion, of course, as you'll recognise. And then the pedipalps in the scorpion are these claws, and the pedipalps in this Europygid are these structures here. Mites as well. These are chelicerates. This is a dust mite, again with chelicerae and pedipalps. And this is an animal that I hope you don't encounter. This is a tick. So these again are chelicerates, but they've retained a remarkably elastic cuticle on the opisthosoma, and they of course drink your blood and expand to enormous size and then drop off. Ticks are a problem, particularly actually in the south of England, where they spread Lyme's disease. So Lyme's disease, a very nasty infection. It's actually a bacterium that gets injected with the tick's saliva, a spirochete. And if you're bitten, you end up with this rather bullseye-shaped scar around the bite site. And it's best treated rapidly if you do. So if you go walking anywhere where there's long grass, these things will jump on you, and it's best to check if you come back from a walk that you haven't got a tick attached to you. Pets can also come in. Our cat came in the other day with a tick. We've managed to dispatch it fairly rapidly. Okay, so that's the chelicerates. Okay, so no antennae, chelicerae, 4 pairs of walking limbs, mostly terrestrial now, but a much richer heritage within the sea. Now there are the myriapods, okay, the second class. These include the centipedes, which are carnivorous, and the millipedes. These are herbivores. Lots of legs, as we've already said, tend to be not differentiated. 2 tagmata, the head and the trunk. They have antennae, mandibles, and 2 pairs of maxillipeds. Like the insects, they lack the second antennae. So they've only got the first antennae, then they've got a gap. And the head of a myriapod is remarkably similar in its construction to the head of an insect. And of course, also they have only the endopod branch of the walking limbs. And it used to be thought that myriapods and hexapods were part of a group, the so-called Uniramia, because of the similarity in the head and in the limbs. And the insects had evolved by shortening of the body of a myriapod and tagmosis, more tagmosis of the trunk. And that no longer seems to hold up. Indeed, it seems that the head of myriapods and the heads of insects have convergently evolved on this same sort of solution. So there's about 3,000 centipede species, about 10,000 millipede species. All of them are terrestrial. Oh, another similarity, of course, between myriapods and insects. Because they're terrestrial, they've had to do away with gills, and in order to do gas exchange, they have these tubes that extend into the body in each of the segments. So they go in through these little holes called spiracles, and these tracheae, they're progressively branching tubes that feed the cells of the body with oxygen. And if you dissect an insect, or if you dissect a myriapod, you can see all these branching tubes inside. And again, this is something which has evolved convergently within myriapods and within insects. So there are some myriapods here. This is a centipede. You can see in the centipede the first limbs of the trunk are these poison fangs. So these are actually trunk limbs that bend round around the head. Centipedes are carnivorous, dorso-ventrally flattened. The legs stick out to the side and are quite long. So these things are built for speed, built for carnivory, built for pursuing prey. Millipedes, on the other hand, tend to be circular in cross-section, so they're much more circular transverse section. The limbs are underneath the body rather than stuck out to the side, and there are 2 pairs of legs per segment, whereas there's only 1 in a millipede, and they have an amazing ability to gain traction with the substrate. So they can grip hold of the substrate very tightly, and they can bulldoze their way along. And indeed, they tunnel through decaying leaf matter and vegetation, and they're vegetarian. They eat decaying vegetable matter, whereas these guys are predators. So completely different mode of life. There's also a couple of other groups within the myriapods. You'll probably never encounter them. But there are also extinct myriapods, giant myriapods, things like Arthropods here. And this is very much the model completely to scale. So it was a predator, probably ate lizards and other large arthropods from the Carboniferous, of course, when oxygen levels seem to have been very high. And there were lots of very large and impressive arthropods around at that time. So here's the front end of a centipede, a scutigromorph centipede. You can see these massive mandibles. You can also see these poison fangs, these spiny, horrible spiny sort of things, needle-sharp things coming from the first trunk appendages. Huge antennae, compound eyes. The head of this scutigromorph centipede looks very much like the head of an insect. Again, convergently evolved, and these lovely great big long running legs. And there are the mandibles there in this close up, and there are the poison fangs coming from the anterior trunk. Racing car design, predatory, carnivorous. A millipede, on the other hand, bulldozer modification, cross-sectional, circular cross-section, and 2 pairs of legs. You can see it here per segment. There are some that have a more flattened appearance, and a huge range in sizes from very, very small to the giant African dewlid millipedes, sort of yay big. So that's myriapods. So just to recap then, and I'll take a short break. So at the base of the euarthropods, possibly pycnogonids. Then you have the chelicerates. Next up, you have the mandibulate groups, the groups with mandibles. The basal group of those mandibulates are the myriapods, and we're now moving into the pancrustacea. We usually deal with those in 2 groups, the crustaceans and the hexapods, but of course it's important to remember that the crustaceans are paraphyletic with respect to the hexapods. So insects, hexapods, are just a specialised group of crustaceans.